plastic has become one of the world's worst pollutants. In the ocean, plastic has a dramatic effect on wildlife. Marine life can become sick after eating it and even die. The oceans are currently estimated to be filled with 15 to 51 trillion micro-sized particles of plastic and is being filled by 8 million metric tons of plastic pollution each year. This amount of plastic is not able to be degraded quickly. It takes about 450 years for one plastic bottle to degrade. What if we could use bioengineering to fix this? While conducting research on microorganisms found from a plastic bottle recycling site in Osaka, Japan, scientists discovered the bacterium Ideonella Sakaiensis, which was able to break down one of the most common plastics in the world, polyethylene terephthalate, also known as PET. PET plastic is formed through the esterification of terephthalic acid and ethylene glycol. Esterification is the dehydration reaction of an organic acid and alcohol to form an ester bond in water. Because ester bonds are strong, they're resistant to degradation. This is one of the main reasons that it takes plastic so long to break down. Ideonella Sakaiensis contains an enzyme called PETase. This enzyme first breaks PET plastic down into monohydroxyethyl terephthalate, or MHET, bis-2-hydroxyethyl terephthalate, or BHET, terephthalic acid, and ethylene glycol. BHET is then broken down by PETase into MHET, terephthalic acid, and ethylene glycol. MHET is then hydrolyzed, meaning broken apart through the addition of water, by MHETase, another enzyme found in the bacteria, into additional ethylene glycol and terephthalic acid. This process may be instrumental in combating plastic waste, and scientists are already jumping on board. A Harvard biodesign team genetically modified E. coli to use in a device they invented called Plastibac. The team inserted the genes that code for PETase and MHETase into the E. coli, and then placed the bacteria into the Plastibac device. The device would float at the surface of the ocean and allow seawater to flow in. If there are microparticles, a filter catches them and then the enzymes degrade the plastic. Then the ethylene glycol is separated into a deposit box, while the terephthalic acid is sent into the microbial fuel cell. A bacteria uses this as a fuel source and produces electrons that are sent to the cathode, creating a measurable voltage, which scientists could use to track the flow of plastic in the oceans. The future of bioengineering bacteria is exciting because who knows what abilities that we will discover. This upcoming science has the potential to make a major impact on plastic pollution and may even save a turtle or two.